Um, my name is Christian Schweizer. Yeah, you know this. So we have our first talk, our first speaker, Chandrasekhar Venkataraman from Sussex, but on the move, right?
Yeah.
Good. We'll talk about image-driven modeling of cell motility, please.
Okay. So I'd like to start by thanking the organizers, both for inviting me to give a talk, but also for putting on this program. It's really been a lot of fun. I've enjoyed all the talks and also interacting with all the speakers. The title, it probably describes 50% of the talks in this workshop, so I wasn't too sure what I'd talk about until very recently, but hopefully this gave me the opportunity to talk about whatever I wanted, basically. But hopefully what I'm going to talk about fits into this general title. So I'm going to try and talk to you about two problems today. The first is an approach that we've worked on with some collaborators to cell tracking, and this is from a mathematical perspective. So if any of you were in Kishore's talk yesterday, we saw some excellent examples of cell tracking and some very fancy algorithms to do it with. This will be much more basic, but it's from a more mathematical perspective. The second problem I'm going to talk to you about is on a growth model for fish and yeast. So Björn Stinner mentioned this in his talk yesterday, and we're going to talk about some slight extensions to the model he presented yesterday. So I think I heard in yesterday's talk that fish and yeast doesn't count as cell migration. So if we don't have time to get to this problem, I don't guess it's such a big deal. So my relatively naive understanding of cell tracking is that basically what we have is a series of static images of a cell. It might be in 2 dimensions or 3 dimensions, and we're interested in reconstructing dynamic data from these static images. So we want 2D plus time or 3D plus time data. We have particle tracking, where you basically want to track things like cell centroids or macromolecules within the cell. So essentially, as I understand it, you want to track a 0-dimensional object. My understanding of this problem is that it really comes down to segmentation. Once you've segmented your particles, tracking them can be quite simple. On the other hand, whole cell tracking is where one wants to recover the whole cell morphology over time. So you have a series of images of the cell boundary, say, and you want to reconstruct the trajectories of points on the cell boundary. And this is important if you want to investigate geometric features like the evolution of cell surface area or volume over time. If you're interested in curvatures, the number of pseudopods a cell has, you probably have to do something like whole cell tracking. Some examples, particle tracking, as I said, you might segment things like the cell centroid and then reconstruct their trajectories by linear interpolation, matching with nearest neighbors, and that can be reasonably straightforward. For whole cell tracking, there are lots of methods in the literature. A lot of them are based on level set type methods, so you basically have 2 curves or 2 surfaces, which describes the position of your cell at 2 distinct times, and you want to compute a trajectory mapping points on one curve to points on the next. There's a very nice method based on electrostatics proposed by Till Bretschneider and some co-workers at Warwick, where if these 2 curves, for example, represented the boundary of your cell at 2 distinct times, you label virtual marker points on one curve with a positive charge, virtual marker points on the other curve with a negative charge, and you compute an electrostatic field between the 2 charged segments, and that gives you your trajectory. So you get a force which basically corresponds to the trajectories of these virtual marker points, mapping one curve to the next. Our approach is slightly different in that we're going to start with a mathematical model for how we think the cell is evolving. And we're going to try and fit this mathematical model to the experimental imaging data. So in that sense, it's more like an inverse problem where the data comes from experiment. So all of this work on cell tracking that I'll talk to you about is joint work with Kostas Blazakis, Anatida Matvamusa, Vanessa Stiles, and Fengwei Yang, who were all at Sussex during this work. Kostas was a PhD student, and he's completed this year and now moved to Greece. And also with Carlos Reyes Aldesoro, he's an image analyst who was at Sussex, and he's now moved to City. I should say that Kostas and Fengwei Yang did all the heavy lifting in the work I'm going to tell you about. So all the computations you'll see were done by both of them. So the problem I want to consider, the basic idea, as I said already, is that we want to reconstruct whole cell morphologies from static images. And as we mentioned, the majority of existing approaches are kind of geometric in nature. So they only take into account geometric considerations. They might want to equidistribute vertices on the 2 different curves or something like that. Our approach, as I mentioned, is slightly different. We want to think of it as trying to fit a mathematical model for the evolution of the cell to the imaging data. And for this talk, I'm going to focus on geometric evolution law type models. So we're going to propose an evolution equation for the cell boundary. And the reason we consider these sorts of models is that they arise very naturally in a number of different models for cell motility. So they're building blocks of all these different works here where people have tried to model cell motility. In many cell tracking scenarios, you don't have information on spatial localization of motility-relevant proteins, so you might not visualize actin or myosin. All you might see is a phase contrast image of the cell or something like that. So all you'd have really in this setting is the position of the cell membrane or the position of the cell boundary. And we're going to work on, in this most basic setting, within this setting we're going to propose a model for the evolution of the cell that's probably the simplest geometric evolution law I could consider. So mean curvature flow with forcing. And the unknown that I'm going to try and find is this forcing, eta, such that the evolution of the solution to this model equation is as close as possible to the experimental imaging data. One way of interpreting this equation is that the normal velocity of our boundary is given by this forcing. We have a term that incorporates volume conservation. That's not necessary for the model, but it's just to show you how one can include constraints like conservation of the enclosed volume. You could also look at things like conservation of the surface area, then one would consider a higher order geometric evolution equation. You could also consider the cell having a preferred curvature, and again you consider a higher order geometric PDE. And you can interpret this term in two different ways. It's either a regularization, which ensures that my curve or surface stays smooth at all times, or you could relate this to a surface tension, and then it has a physical meaning. So H is the mean curvature, by which I mean the sum of the principal curvatures. So the problem I want to consider for most of this talk is to find a forcing such that the solution to this model equation and the experimental imaging data I have are close in some sense. And the reason I've put the inverted commas around close is that I'm not sure how best to define close if I formulate the problem like this. So if we formulate the problem like this, what we're trying to do is the optimal control of the sharp interface formulation of a geometric evolution law. And at least as far as I'm aware, there's not a lot of mathematical theory for this type of problem. So Björn mentioned in his talk yesterday, it's not obvious how one defines a smooth notion of closeness between the different surfaces. What regularity would we want on this forcing function and so on? Many of these problems remain open. So to try and get around that, I'm going to reformulate my problem. Instead of working with sharp interfaces, I'm going to work with so-called diffuse interfaces. So the idea here is that you take your sharp interface and you thicken it a little bit. For example, if the boundary of my cell was described by this black curve here, and this black curve was contained in some two-dimensional domain omega, I would compute a signed distance function to this curve. So every point x in omega, d gamma x gives you the distance to the closest point on the curve, and it's negative if you're in the interior of the curve and positive if you're outside the curve. Then, given these two things, I can compute a diffuse interface representation of the curve, and here the thickness of the diffuse interface would be 2 epsilon, if I computed it according to this formula. And basically, the diffuse interface would be such that this function phi took the value -1 if you were more than epsilon away from the curve and inside, +1 if you were more than epsilon away from the curve and outside, and you smoothly transition between -1 and +1. The 0 level set of phi gives you your curve. You can do this if you have multiple cells as well, but the nice thing is that you only need a single diffuse interface phi. So if I wanted to track multiple cells, I don't need to segment each cell individually. I just have one diffuse interface function or phase field function phi, and that can describe all the different cells, assuming they're sufficiently well separated. You may not even want to actually segment your cells in the first place. If you have data of sufficiently high contrast such that you can just threshold or something like that, you already have a diffuse interface representation of your cells. So in that setting, you don't even have to do any further segmentation. You could just work directly with the raw data. If we adopt this diffuse interface approach, then the corresponding approximation to the force mean curvature flow equation is the so-called Allen-Kahn equation with forcing. So here G is a double well potential, such as this quartic potential here, which has minima at plus and -1. And there's a huge amount of mathematical literature on the convergence as you take epsilon to 0. So the width of the diffuse interface to 0 on the 0 level set of the solution to this equation converging to a solution to motion by mean curvature. We had this lambda, which in the sharp interface case was a constraint on the volume enclosed by the surface. In the diffuse interface setting, one can interpret this as a constraint on the mass of phi. And as you take epsilon to 0, you recover a constraint on the enclosed volume. So to give you an idea of what the problem is now, let's think we have a single image that we want to fit to, and we have a diffuse interface representation of that. And we have an image at a previous time, and I use that as initial data for this PDE here. Then my cell tracking problem can be formulated as a PDE constrained optimization problem, where I want to minimize this objective functional here, which consists of two terms. I have a term which is a so-called fidelity term, which measures the difference between the solution to my model and the desired data. And I have a regularization term, if you want to think of it in that way. And so gamma is some small positive parameter, and this term is needed to ensure that I obtain a well-posed problem. I want to minimize this objective functional subject to the constraint that 1 holds. So this is a PDE constrained optimization problem. But the nice thing about this problem here is that it's just a semi-linear parabolic equation posed on a time-independent domain. So there's a huge amount of mathematical literature for the solution of these types of PDE constrained optimization problems. There's also been some nice computational work. So Axel Voigt, together with Hauser and Rascher, have looked at a couple of phase field formulations of free boundary problems and tried to solve optimal control problems with those as the state equations. So how would one solve a problem like this? Well, you can go to your favorite textbook on PDE constrained optimal control problems, such as this book by Troelsch. You introduce, one way of approaching the problem is to introduce a so-called Lagrangian or function L, where you've introduced P, which you can think of as a Lagrange multiplier that ensures the state equation holds. And we then formally derive the so-called first order optimality conditions. This has not yet been made rigorous. Stationarity of the Lagrangian with respect to P gives you your state, and these are the remaining two first order optimality conditions. The first one gives you your so-called adjoint equation, which you basically use for efficient computation of the derivative of the objective functional, and you get this equation that characterizes the optimal control eta star. With all of this in hand, we propose a very simple algorithm for the resolution of the optimal control problem based on a steepest descent method. And the basic idea of the algorithm is that we compute the solution to the state equation with the corresponding control eta. We had this constraint on the mass that we had to impose, and there's a very nice algorithm proposed by Blowy and Elliott for enforcing this constraint. Once you've computed your state, you compute the objective functional corresponding to the state phi n and the control eta n. If you meet the tolerance, you exit. If not, you carry on and compute the adjoint state Pn, which is basically used for efficient computation of the gradient of the objective functional. But if you look back at the adjoint equation, this was a linear parabolic equation posed backwards in time, but the state enters the adjoint PDE. So computing the adjoint state P, which we have to compute backwards in time from the end time back to 0, requires the computed states. So we basically have to store the states as we solve this algorithm forwards in time. Once you've computed your adjoint equation, you update your control according to the gradient. Then n plus 1 goes to n, and you keep iterating until you meet your tolerance. This is a very expensive problem computationally. As Axel mentioned in his talk, if you're trying to solve these phase field equations in 3D, for example, grid adaptivity is mandatory. And Fengwei Yang is an expert on the implementation of parallel adaptive multi-grid based solvers for these types of equations, and he's implemented a very fast numerical method for solving the state and adjoint PDEs. We also had another idea, and we're calling this for now the two-grid strategy. And basically this idea is based on the observation that while the phase field equation is a semi-linear PDE, the adjoint equation here is a linear parabolic equation. And it's kind of well known that you need a lot of resolution in the interfacial region for the solution of the state equation forwards in time. But perhaps because the adjoint equation is linear, even though it has a coefficient which varies a lot in the interfacial region, perhaps because it's linear we can get away with a much coarser grid for the solution of the adjoint equation. So this is what we've tried, and at least numerically it seems to work quite well. So we use an adaptive grid for solving the forward problem, and then we solve the adjoint equation on a coarse uniform grid. This obviously gives you better efficiency because you're using fewer degrees of freedom for solving the adjoint equation, but it also gives you massive memory savings because I have to store the state and when I solve the adjoint equation, but now I only have to store it on this coarse uniform grid. I never have to store it on the adaptive grid, and it's also easier to implement because if I was working with an adaptive grid, I'd need to solve backwards in time on the same adaptive grid. The memory savings are a big deal. For example, using this strategy, you can solve these problems in 3D on a desktop or even on a laptop, whereas you'd only be able to approach them in 3D in parallel if you're using adaptive grids for both equations. We also have a very simple algorithm for an adaptive choice of the step size in the steepest descent based update, and that seems to give better efficiency as well. But for this talk, I wanted to focus more on what the method can actually do and to see if it would be any use to practitioners. So here's one very simple example. I think the images come from images of zebrafish neutrophils in vivo. And what I'm showing you here is the zero level set of the phase field function phi computed with the optimal control. And it's shaded by the value of the forcing on the curve. So what you see is that you generate large protrusive forces. Let me go back. Towards the front of the cell, and you have contractile forces towards the cell rear, and at the final time you're very close to the desired state. To illustrate why constraining the volume might be a good idea, in this example we have the same initial data and the same target data, but on the left we have no constraint on the enclosed volume, and on the right we have this constraint on the volume, which we interpreted as a constraint on the mass. And what you see is that on the left you lose a lot of volume midway through the evolution, whereas on the right you keep roughly, well, you don't conserve exactly the volume because our constraint is actually on a time-dependent volume, which is a linear interpolation of the volume enclosed by the red curve and the green curve, but you don't lose too much volume during the evolution. Okay. Maybe that shape was a bit too easy to try and track, so we took some more data, which came from this paper here, which I think was in cytoskeleton, I think this is a epithelial cancer cell that's quite invasive, and so you see it generating these very elongated protrusions at the back, these long tails, and they're quite fine-scale features. We took a few different frames from this evolution. The algorithm can be extended straightforwardly to multiple frames, and we tried to track it. So what you'll see here is the zero-level set of the phase field function phi computed with the optimal eta. That's the red curve, and the white guys are where I want to be. So every time my desired state changes, I update the position of the white region. So what you'll see is that we seem to track the white regions quite well. But if you look closely, you'll notice one issue with this method in that our curve changes in topology. So we have these splittings where we leave behind a bit of cell. That's perfectly admissible within this framework. If you work within a diffuse interface framework, your interface is allowed to change in topology. And clearly for many applications, that's not what you desire. We don't expect that the cell is changing in topology as it's migrating.
Sometimes it does. Might do.
It may do. You'd like to have that as an option, though. You'd like to make it possible that it didn't. But yeah, sure.
I have a question. For each fit, you get an estimate for the model parameters?
No, so we're not trying. So I should. If we fit to one frame, it's kind of it's what I described earlier. We're trying to find a function eta which is defined over space and time, yeah, such that our evolution at the end time is close to where we want to be. So when we're not estimating any parameters, we're actually trying to find this function, if you want to think of it that way. Yeah.
But, I mean, to a first approximation, the Khan-Hilbert or
Alan Kahn.
Alan Kahn implement some kind of tension along the contour.
Yeah.
So you basically, with this eta function, you basically say how much you have to go away from a circle in some sense.
Yes, so if theta was 0 with volume conservation, you would relax to a circle with the correct enclosed volume. So we haven't yet worked out how to fix topology, but one can track changes in topology, which I think still may be useful. So it's by the Gauss-Bonnet formula for a smooth oriented compact or closed surface. The integral over the surface of the Gauss curvature is a topological invariant. And there's some very nice work by Du, Liu, and Wang where they proposed a diffuse interface approximation to this quantity here, which is called the Euler number. So it's defined in this way here. You don't need to worry about what's inside. It's just a diffuse interface approximation basically to K. But you integrate only over the interfacial region. So you pick parameters B and A, and they even give you a methodology for picking these parameters. B is strictly greater than 1 and negative. A is strictly less than 1 and positive. And so this integral over omega AB is only over the region in which the diffuse interface is. You don't integrate over the -1 and +1 phases. And if we're working in you have a formula in 3D as well, it's a little bit more complicated. If we're working in 2D and our cells are all simply connected regions, then this guy here is approximately equal to the number of cells in our image. So each time this changes, it means we've added a new cell, or lost a cell. So using this, I'll show you in a second that we can track changes in topology. We haven't yet worked out how to fix topology, and we're currently trying to understand how one might penalize changes in topology. And there's some very nice work by Dondo, Munyai, and Roger in that direction. Yeah.
How much does that have to change to count a new cell? Because you're going to get a continuous result out of that.
Yeah, you'll see. So actually, that's a good question. So in that paper, they try. I mean, it gets a little complicated, but they also have an interpretation that says, as you're passing through singularity, you get. So this is an integer if your phases are simply connected regions. But as you pass through the singularity, they have a fractional interpretation of the Euler number as well. So, but you're right, it is continuous. So to show you one example of how this works, we got this data from YouTube. That's why the quality is quite poor. Forget about these cells in the bottom right-hand corner. We're going to ignore them because they're too near the boundary. But what you see in the data is that initially this cell divides, and then these two will divide right near the end of the simulation. So we started out with 3 cells, and by the end we have 6 cells. The algorithm will work fine to try and track this. Cell division is no problem. So we picked a few frames from this video, and we tried to do the cell tracking. And the results you get out are here. We started with 3 cells initially. 1 divides, and then towards the end of the simulation, the next 2 both divide almost simultaneously. And if we track this diffuse interface approximation of the Euler number, it's initially 3 when we had 3 cells. The first division, it jumps to 4, stays roughly around 4, and then right at the end, it jumps up to 6. So it seems to be reasonable, at least for this one example. All of the approach works in 3D, but the difficulty for us actually is getting good data in 3D. So you need quite high resolution data that describes the cell boundary well, and we haven't yet found any good real data. So the 3D example I'm going to show you is artificial. We started with this sort of cashew nut or crescent shape, and we just rotated it and translated it. So this is our initial state, and this is where we want to get to. And as you can see, we do quite a good job. You get very close to it. I can show you a movie of the evolution as well.
What kind of data do you need? Because you say there is no good data out there.
Well, I think the issue is in the data I've seen, and this may well be wrong, that you have very good resolution in X and Y, but to do this in 3D, you'd want good resolution in Z as well. Yeah. Otherwise...
That's optics.
Sure.
It's physics.
But I guess it would also be what application do you have where you need this done in 3D. So unless there exists an application where you're interested in 3D cell tracking, then there may be no need for it.
You see, I mean, most microscopy techniques are worse by a factor of 3. In the Excel. Is this already so bad?
I don't think, I mean, probably not. I mean, there probably is, even with the 3D data you could give us, the algorithm would generate something. You just wouldn't have very good resolution in one of the directions. But in principle, you could still use this approach. And
sure, if it's not in the data, you can't make it.
Sure. I mean, you can, but
you better not be.
Yeah. So the second problem I wanted to talk to you about was some very brief discussion on a growth model for fish and yeast that was attempting to go a bit further with some of the modeling that Björn mentioned yesterday in his talk. So fish and yeast, as I understand it, it's a model cell type for studying cell polarity, because they're bone polar. They're bone when a parent cell divides, and the old end that existed prior to division is the cell's front initially, and it just grows from this old end after some critical point in the cell cycle, this monopolar growth, where it's just growing from one end, switches to bipolar growth, where it grows in an oscillatory fashion from both tips. And you can see bipolar growth in this video here. Growth of a tip is correlated with the concentration or intensity in these fluorescence plots of CDC42 at the tip. So the hypothesis is that if it's above a certain threshold, then that tip grows. And there's some very nice work in Science by Bass and co-workers where they did some experimental observations of fish and yeast. They saw initially you only have CDC42 at one of the tips, very little at the other tip, and you have growth only from that tip. Afterwards you have oscillations in CDC42 at both tips, and you have growth from both tips. So they also did some modeling work in their paper in Science, where they had a nice model, but reasonably simple, where they had an ODE model for the mass of CDC42 at both tips, so two ordinary differential equations. They didn't model growth explicitly, they just had a time-dependent cell volume, which increased over time, and that entered their ODEs as a parameter. So the ODEs look like this. You have the rate of change of CDC42 at each tip is given by recruitment from a shared bulk cytoplasmic pool and decay back into the cytoplasm. And the volume enters in this way because their assumption is that the tips have a fixed surface area as the volume's increasing over time. And so they propose this model for recruitment. This function lambda is an increasing function of the concentration with saturation, and they show and you also have, the total amount of CDC42 within the cell is proportional to the cell volume. And they show numerically that by choosing this lambda appropriately, initially the system of ordinary differential equations has a single asymmetric steady state, which has elevated CDC42 only at one tip, corresponding to monopolar growth. And as the volume increases, you switch to a symmetric steady state, and you have elevated CDC42 at both tips, corresponding to bipolar growth. But what we saw in the experiments was that you don't have a steady state of elevated CDC42 at both tips. You have oscillations in CDC42 intensity. And they could show actually that you can't get the oscillations out of their model as it was, and to get oscillations they introduced this delayed negative feedback. So the decay rate was enhanced by the value of CDC42 at some previous time. So tau is some strictly positive time delay. And if you implement that, then you get exactly the type of oscillations you see in experiments. This was when we were approached by some biologists who were at Warwick, the group of Graham Lads, he's now at Cambridge, and they were very interested in fish and yeast. And in particular, they were interested in a GTPase in the fish and yeast cell called GAP1. So Graham's an expert in receptor cell biology. And what they'd noticed is that if you knock out GAP1 from these yeast cells, they never go into bipolar growth. They just exhibit monopolar growth. They looked molecularly whether GAP1 affects CDC42, and it appears not to. But on the other hand, they believe that GAP1 downregulates RAS1, which was another GTPase in fish and yeast. And the hypothesis was that created some sort of a negative feedback loop, and that was what was allowing you your oscillations and your transition into bipolar growth. So we did some modeling with them. We settled on a 4-species system where we took into account RAS1, SCUD1, CDC42, and GAP1, and they're in two states. They're either active, which means they're resident on the cell boundary, or inactive, which means they're present in the cytoplasm. To model their dynamics, we proposed a 4-component surface reaction diffusion system posed on the evolving boundary. So this is similar to the equations that Björn was discussing in his talk yesterday. And the reaction kinetics for each species are this form here. So we have recruitment from a bulk pool of each species that is enhanced by or activated by one of the other species. And we have decay back into the bulk. I'm going to assume that on the timescales I'm interested in, bulk diffusion is fast, and so I can treat the bulk species as spatially constant. And the modeling is such that the total amount, bulk plus surface, is conserved. I'm also going to assume that the diffusivities on the surface are small. And the nice thing about doing this is that if you initialize concentrations localized to the tips, they stay localized to the tips for all time. So I only need to define the tips implicitly through the initial data for my surface PDEs. We don't need any explicit definition of what constitutes a tip.
But they are small compared to what?
Small in a non-dimensional sense. So on the timescales you're interested in, you don't have diffusion out covering the entire, you don't have diffusion away from the tip.
Means zero.
Pardon?
It means zero.
No, not zero.
Yeah, but in the model.
No, in the model they're not. So there's diffusion on the surface.
Okay.
But if diffusion is fast, so what I'm saying basically is that if you have initial data at a tip, on the timescales of growth that you're interested in, you don't have diffusion taking that concentration away from the tip. I think it's reasonable, to be honest, at least we need to do more work on this, but I think it's physically reasonable as well to assume that. So in the modeling, we assume that GAP1 downregulates RAS1, so the kinetics for RAS1 are slightly different. The decay of RAS1 is enhanced by GAP1, and that provides you your negative feedback and allows oscillations, as we'll see in a second. So Graham and his colleagues had identified GEF1 as being responsible for the transition from monopolar to bipolar growth. If you got rid of GEF1, even if you still had GAP, these cells would only exhibit monopolar growth. And we're still waiting on spatial localization of GEF1. So for now, the transition from monopolar to bipolar growth is just via a switch, a time-activated switch. To get the period and the amplitude of the oscillations right, we just did some fitting of these phenomenological Hill coefficients in the reaction kinetics. We have an evolution equation for the motion of the boundary of the cell. It's very simple. We're just going to say we have protrusive force if CDC42 concentration exceeds a given threshold, and I have small surface tension and bending rigidity, which one thinks of as regularization. So what do you get when you simulate such a model? If you simulate the wild type, initially you get monopolar growth, so CDC42 is only elevated at one tip. Then at some point GEF1 appears and you transition into bipolar growth, and naturally this bipolar growth is with oscillations. So there are no time delays or anything like that. The aspect ratio is a bit shifted on the projector, but these cells are more elongated than what you see here. If you look at the delta gap mutant, so remember this is what they had data for, and if you knocked out gap, you only see monopolar growth. And indeed that's what we see if I run the simulations, and that's kind of obvious because GAP1 is providing you your negative feedback. If you knock it out, all the CDC42 you've recruited here can never go back into the cytoplasm, and so there's nothing to be recruited at the other end when you add GAP1. So at least for these two very simple cases, it's consistent with what they see in experiments. A very natural criticism of this is why did we bother with a PDE model? Have we learned anything? We could have just considered ODEs, so the zero diffusion case at both tips. And to be honest, for the two cases I presented, that would be fine. We could have had a system of 5 ordinary differential equations for each tip. But there are many other mutants where spatial variation becomes important. So you can knock out RAS1 and these cells round up completely. They no longer have well-defined tips. And we can do that within the framework I'm talking to you. But more interesting, I think, is if you look at other types of mutants, you can just about see it here, where you include pheromone, which is like a chemoattractant for these yeast cells, then you can generate very strange morphology. So you can get these T-shaped cells, which nucleate an extra tip. And if you wanted to investigate something like this, you'd have to have a spatial model. The ODE model wouldn't be able to explain something like this anymore. So that was pretty much everything I wanted to say, just to sum up. We proposed a model for fish and yeast growth that, at least qualitatively, was based on experimental imaging data, and we were able to generate oscillations without time delays. And simulations of the model equation gave results that, again, at least qualitatively, were consistent with experimental data. There's lots of fitting and lots of refining of the model that we plan to do. In terms of cell tracking, we proposed an algorithm for whole cell tracking that one can think of as fitting a mathematical model for the evolution to imaging data, and we've implemented a robust and fast numerical method for solving the resulting optimal control problem, where we used a 2-grid strategy for the solution of the forward and adjoint equations, which is particularly important in terms of memory requirements. I also think this approach could be relevant for general optimal control of semi-linear parabolic PDEs. This could be an interesting method to try and tackle them. As I showed you in some results, we can apply this framework to the tracking of multiple cells. A nice aspect of this approach is that you don't have to solve this so-called matching problem where you match one cell to the next one. This is done implicitly by the algorithm, and it works in 2 or 3 dimensions. One possible application may be if you want to try and estimate the rates of cell division within a population. And with that, I'll stop and thank you for your attention.
Thank you very much. We have time for questions, please.
Can you just go to 4 dimensions with this algorithm? I mean, add time as a 4th dimension? Because you can easily cope with topology changes.
Yes.
So you would have fission and fusion.
I'm sorry, I don't think I
If you take a time series
Yeah.
of three-dimensional shapes,
Yeah.
you will have a four-dimensional problem.
But that's what.
That's what we have. We have a
okay.
series of three-dimensional
So it's-
snapshots.
Okay.
Yeah, exactly.
Yeah.
So these are evolving in time. In
Yes.
3D.
So it's really four-dimensional.
Exactly.
3 plus 1. Okay.
That's right.
One short question. I mean, the segmentation. This is like segmenting with elastic contours, basically.
So I haven't discussed segmentation at all in this. I mean, you don't need the contour explicitly. So for example, if you had a data set for which you could threshold, so let's say the background was at a much lower intensity than the cell body, that in itself is a diffuse interface. You could use that. So you don't need the contour. You don't
I
need.
mean, what you do is segmentation. I mean, your contour is a segment.
Yeah. What you've shown is effectively.
So that is the. So what I'm computing is a diffuse interface. I'm taking the zero level set of that function, but actually what you compute is not that. Yeah.
But what you do is effectively you also
Implicitly
get a
you get it.
segmentation.
Yeah.
Sure, but that
How it compares to this elastic contour.
But it's not
segmentation.
really. Yeah.
Is it faster than the elastic contour?
it'll be slower.
Another question, please.
For your problem of topological changes, I think, well, of course you can constrain the number of objects seen, but I think there might be more mechanically based ways also to do that.
Yeah.
And I think one thing that could be improved is adding some anisotropy, either a global one at the cell scale, where you, because if you go back to the slide where you have the cell migrating, there's one. So you have the migrating one, the single cell migrating. Yes.
Yeah.
So you actually have in the center part of the cell, you have the fibers which are aligned with the cell orientation also.
Yeah.
And so most of the stress is aligned in that direction, and the protrusivity is also only at the ends. So if you add some bias, some penalty to deformations in one direction relative to the other, that could be very simple at the global scale. Or if you have edges which have different properties, which can move more normally than others, then you may solve the problem with something which is closer to the actual biological problem. Constraining the number.
So I guess what you're saying is that the model for the evolution is much too simplistic and needs a lot more biologic, which I completely agree with, and something like that sounds like a very reasonable way to improve. Yeah, sorry.
I noticed you always had a big strip around the periphery for your representations. Does that represent somehow the width of your diffuse interface?
So as you take epsilon to zero, the width of that strip will get smaller.
Yeah, okay, but that's, you could have just put a curve down the middle of it, and that would have been the interface.
Yeah, so the interface is the zero level set. I mean, your representation of the interface is the zero level set.
Another question? Then thank you again.
Thank you.